Hello, everyone. I hope you had a chance to watch the Lady Tigers play in the national tournament. Didn't turn out the way we'd hoped, but that's the way it goes sometimes. There's always next year, I guess. So, when we left off on Wednesday, I was going over the different parts of the nephron. The nephrons are tiny microscopic filtering units that filter the blood and form the urine. And the main parts of them are the renal corpuscle, which includes the glomerular capsule and the glomerulus. Then there is the proximal convoluted tubule, the descending limb of the nephron loop, and then the ascending limb of the nephron loop, and the distal convoluted tubule, and the collecting duct. From the collecting duct, the urine is released at the renal papilla, and then it travels through the passageways of the kidney until it is released into the ureters, which carry the urine down into the urinary bladder. So now that we've reviewed the nephron, let's take a closer look at the renal corpuscle. Okay, here's the renal corpuscle. So the renal corpuscle is composed of the glomerular capsule, which has an inner and outer layer, and in between is the capsular space where the filtrate passes into after it goes through the filtration apparatus. And then from there it goes into the tubular portion of the nephron. So, the glomerular capsule has an outer layer made up of squamous cells that's called the parietal layer. Then there's a hollow portion, and then there's a deeper layer that's called the visceral layer. The visceral layer is composed of podocytes, which have cytoplasmic extensions that make contact with the capillaries of the glomerulus, and they surround the glomerulus, and adjacent feet of the podocytes, which are called pedicels, have interdigitations, where the little finger-like processes that interdigitate, just like you can see on my fingers, my hands, and it leaves very, very thin slits between these interdigitation. These are called filtration slits, and those actually form the very finest filter that prevents large substances like blood cells and proteins, larger molecules, from passing through the filtration apparatus. And so instead, those large things like blood cells and proteins stay in the plasma of the blood in the glomerulus and don't get filtered. Because if they got filtered, they might end up getting lost by being excreted in the urine. All right, so let's move on then. So what we're going to do next is talk about the major processes that are involved in urine formation. So there's basically 4 different processes. The first is called filtration. Filtration occurs in the renal corpuscle when the hydrostatic pressure of the blood forces some of the plasma, minus most of the proteins and blood cells, into the capsular space, the hollow portion of the glomerular capsule. Then from there, the filtrate goes into the tubular portion of the nephron. Now, in the tubular portion, there's 2 processes that occur. The first is called reabsorption. Reabsorption is the active transport of useful substances like electrolytes and water from the tubule into the peritubular capillaries that surround the nephron. Then in more than one part of the tubule, there's also a process called secretion that occurs. Secretion is basically just the opposite of reabsorption. It's the transport of waste products and toxins and drugs that did not get filtered from the peritubular capillaries into the filtrate that's in the lumen of the tubule. So that gets rid of toxic substances that may not have gotten filtered. And so those toxic substances can then pass out with the urine. And then finally, the fourth process that results in the production of this concentrated waste that we call urine is water conservation. So water conservation actually occurs in several different parts of the tubule. Most of it actually takes place in the proximal convoluted tubule. There's additional water conservation or reabsorption takes place in the nephron loop. And then depending on the hydration status of the individual, there may or may not be more water reabsorption that occurs in the distal convoluted tubule. That reabsorption that occurs in the distal convoluted tubule is under hormonal control. So together, those four processes then result in the production of urine. So what we're going to do in most of the rest of this chapter is to go through each one of these processes and take a look at the molecular mechanisms that cause these processes to happen. Okay, this is a close-up illustration showing the filtration apparatus. So on the far left, you have the lumen of the glomerulus. That large red structure you see on the lower left is a red blood cell, just to give you an idea of the size. So notice just to the right of that red blood cell, you can see a cross-section through the wall of the glomerulus. And notice there's some holes in that wall. Those holes are called fenestrations. Those holes prevent blood cells from being filtered and escaping the glomerulus. They don't prevent smaller molecules from passing through the wall of the glomerulus, however, because there's also two other layers of the filtration apparatus. The next layer, which is shaded kind of a bluish color, just to the right of the glomerular wall, is the basement membrane of the glomerulus. The basement membrane is composed of proteoglycans, a combination of protein and carbohydrates that form a finer filter. And the basement membrane will only allow molecules smaller than about 8 nanometers to pass through the filtration apparatus. So that prevents most proteins, all but the very smallest peptides, from passing through the filtration apparatus. Then finally you have the slit pores, which are the thin spaces between the pedicels of the podocytes. Those slit pores only allow molecules that are smaller than 3 nanometers to pass through the filtration apparatus. So that's actually forms the very finest filter. So the filtrate that passes through the filtration apparatus is similar in composition to the plasma of the blood, minus most of the proteins. The filtrate only contains about 0.03% proteins compared to the 7% that's found in the plasma. Plus, it contains water, it contains electrolytes, small nutrient molecules like glucose, amino acids, fatty acids, vitamins, and waste products such as urea, uric acid, and creatinine.
On blood and urine composition if the filtration membrane is damaged or destroyed.
I took it from source, so audio is not very good, but you get an idea of kind of how the filtration apparatus works. So down at the bottom of the photomicrograph that you see here, those large black areas are actually the red blood cells, and surrounding them is the plasma that's in the glomerulus, And then above that, you have the filtration apparatus. So if you watch this closely when I start the animation, you'll see large molecules like large oval structures bounce off the filtration apparatus because they can't pass through, but smaller molecules can go through and enter the capsular space.
Blood and urine composition if the filtration membrane is damaged or destroyed.
So those large purple ovals, notice they're bouncing off, they can't go through. Those little tiny purple spheres can go through the filtration apparatus. Okay, so that kind of gives you an idea how it works. All right, now, next topic in this chapter are the forces that drive filtration. So the main force that drives filtration is called the blood hydrostatic pressure. Blood hydrostatic pressure is basically just the blood pressure. It's the pressure that's produced when the heart forcibly expels the blood from the heart into the arteries. It's forced out under pressure. Once the blood gets to the glomerulus, the pressure of the blood is only about 60 millimeters mercury. Okay, so that's what forces most of the plasma out of the glomerulus and into the capsular space. Now, in addition, there are two opposing forces that oppose the filtration of the blood in the renal corpuscle. The first is called colloid osmotic pressure. Colloid osmotic pressure is caused by the fact that the plasma of the blood in the glomerulus has a much higher concentration of protein than the filtrate that has passed through the filtration apparatus. So since there's more particles of protein in the plasma, water tends to move from the filtrate back across the filtration apparatus and back into the blood of the glomerulus. And so that colloid osmotic pressure actually exerts a force of 32 millimeters mercury in the opposite direction that the hydrostatic pressure is pushing the filtrate. In addition, there's also a capsular pressure caused by the pressure of the fluid that moves into the capsule. All fluid exerts a pressure on the surrounding walls in which it's contained, and that filtrate, when it goes into that hollow space of the glomerular capsule, is no exception. And so that fluid exerts a pressure of 18 millimeters mercury on the surface of the filtration apparatus. That kind of pushes some of the filtrate back into the blood in the glomerulus. So if you add together the two opposing forces, the colloid osmotic pressure and the capsular pressure, that comes to a total of 50 millimeters mercury. That's only 10 millimeters lower than the hydrostatic pressure that's the main driving force for filtration. So there's really not much of a pressure differential that's needed to cause filtration. But that's low enough that if there's a drop in blood pressure to the glomerulus, that could very much reduce glomerular filtration. And if glomerular filtration stops, then the blood is not going to, the waste products in blood are not going to be removed from the blood. And urea will build up into the blood until it becomes toxic. All right. Now, next topic is called glomerular filtration rate. This is a measure of the amount of blood that's filtered by both kidneys per minute. So the glomerular filtration rate for males is 125 milliliters per minute, which doesn't sound like a whole lot, but that actually amounts to 180 liters of blood that's filtered per day. That's actually quite a bit of blood. GFR for females is a little bit lower. It's about 105 milliliters per minute. But again, that amounts to about 150 liters of blood per day. And so your glomerular filtration rate must be very precisely controlled, because if blood pressure drops just a little bit to the kidneys, then GFR will decrease and the kidneys won't be able to get rid of all the waste from the blood. If GFR is too high, like if a person has high blood pressure and GFR is not regulated, then you're going to be producing a lot more filtrate. There's a greater chance of losing fluids and electrolytes in the urine. And so it's really important that GFR be regulated very closely. And so there's several mechanisms that do that. One class of mechanisms is called autoregulation. And that's just a regulation of glomerular filtration by the afferent and efferent arterioles themselves that supply the nephron with blood. And then there's another more complex mechanism that's called the renin-angiotensin-aldosterone system. That's a hormonal mechanism of regulation that involves several enzymes as well as a hormone aldosterone. That's a fairly complex mechanism, and so I think it's probably best if we just stop here and then pick up after we get back to class next week. So, not sure because of the weather forecast whether we'll have class on Monday or not. If we don't, then we'll pick up again on Wednesday. If the weather forecasters are wrong and the precipitation comes down as rain, then I'll probably see you again on Monday. So, hope you have a good weekend, and make sure you work on your Connect assignments, and make sure you read your textbook, and make sure you study, and I'll see you next week.